Okay, so I'm very happy to introduce Amy Kosieski, who's the next speaker, and we'll talk about the Krakencoder, which is a multimodal, multi-atlas connectome mapping and fusion tool. Amy is joining us via Zoom from Cornell, and we're very glad to have her here in this way. Thanks so much.
Great, thank you for having me. Yes, so today I'm going to be telling. Oh, is that? Do you hear the feedback, Keith, or is it just me? Maybe it's just mine. Okay,
I heard it for a second, but
yeah. All right, let me try again. Hopefully this will be okay. All right, so today I'm going to be telling you about the Krakencoder, and as Ariel so kindly told me, for the Seattle natives out there, it's all it's kind of on theme, because we do have I guess you guys have a ice hockey team, I think it is, that is the Kraken. So I just wanted to kind of put that one out there, that I'm glad to be presenting to a group of people in Seattle. All right, so today, sorry, 1 second. I'm just trying to get, I want to be able to see you all. And for some reason, when I do that, it's not letting me see you guys. So let me just see if I can keep this up there. Okay, so let me go. Okay, that's better. All right, so today you're going to be learning about, and all week, the next couple of weeks, I'm sure a lot of you are going to be picking up things on mapping the human brain with MRI. Maybe a lot of you know about this already, but let me just go ahead and kind of get everybody on the same page. When we measure the brain with sort of MRI and neuroimaging, the two modalities that we tend to use in our group are diffusion MRI and fMRI, mostly resting state fMRI. And so diffusion MRI is this modality where you have sort of tracking of the diffusion of water molecules through the brain, and that can allow you to sort of reconstruct likely white matter pathways that connect different parts of the brain. So it kind of gives you the backbone or the wiring of the brain. And then on the other side of that, we have the functional sort of time series activation that occurs in the brain over time while a person is, told to sit there in the scanner, not think about anything particular, or if they're told to do some kind of task, then the MRI scanner kind of picks up on this blood oxygenation level dependent signal, and you get this time series that's tightly coupled to neural activity, hopefully. And so from the different parts of the brain, if we kind of chop up the cortex and the subcortex into parts, then we can reconstruct a graph or a connectome that allows us to sort of capture the pairwise strengths of connections between different parts of the brain. So in structural connectivity terms, it's sort of how much wiring there is between pairs of regions. So that's kind of what we put in the entry of the structural connectome matrix, is how many sort of connections there are, or counts of streamlines that connect different parts of the brain. And then functional connectivity, on the other hand, is sort of how correlated in time the activation time series are for two different pairs of brain regions. And so from that, we get these networks or graphs that sort of capture overall brain networks, both structurally and functionally. And here you can see sort of this sped up movie on the lower right-hand corner of different sort of pairs of regions of the brain and the streamlines that connect them, and then the sort of corresponding entry in the structural connectivity matrix. So a lot of people have been working, including my lab, on looking at the relationships between structure and function. So you might think that if two sort of brain regions are wired together, that they would fire together. And that's generally true. But if you just look at, say, a correlation of the structural and functional connectivity matrices, and you take the upper triangular part of it, you vectorize it, and you correlate it, you usually get values at 0.25, 0.3. So they don't really correspond that tightly to one another. And if you think about it, you probably don't want them to correspond so tightly together, because your brain should be a dynamic system that sort of responds to incoming information and different can attend to different things and complete different tasks. So we can imagine that we might want to have a sort of dynamic repertoire that our brain can achieve, rather than just relying on those structural connections that are there in the brain, that are sort of wired and they're sort of hard-coded in some respects. So people have been looking at using different kinds of modeling techniques to map structure to function. So there are things like neural mass models, which are sort of systems of differential equations that describe the activity over time of different parts of the brain. And then you can add in on top of that structural connectivity information to allow different parts of the brain to communicate with one another via a spread of activation along the white matter fibers. Then there's things like statistical modeling, where we just sort of look at the straight relationships and try to do maybe a linear regression of predicting one modality, perhaps functional connectivity, from different topological or different kinds of properties from the structural connectome. And then there's a whole host of things that are using graph theory. So sort of things like network diffusion or message passing or communication strategies based around representing the brain as a network. And then applying sort of graph theoretical properties to understand how activation or functional activation might flow through this network. And then today I'm going to be telling you more about, I'll start with a little bit with network control theory and tell you about some of the work that my lab's been doing using sort of biophysical models of dynamic systems. And then I'll talk about the Krakencoder, and mostly at the end, second half of the talk will be about the Krakencoder, where we just kind of throw out all of our sort of a priori knowledge, biophysical knowledge, and we kind of do a data-driven approach to map between structure and function and structure and vice versa. And so one of the sort of motivations for looking at structure-function relationships is that there's been a lot of work in investigating how structure and function relate to one another and how in disease or recovery. And this includes neurological and psychiatric disorders. So two of the studies I want to highlight had used the virtual brain framework. That's a system of neural mass models that simulate functional activity at each region, linked by the structural connectome. And those sort of two studies showed a correlation between the model parameters from the virtual brain that were found by fitting the model to the data. And they found that there was a relationship between those parameters that they extracted from the, that they used that they fitted the data on, and cognition in Alzheimer's disease and motor impairment after stroke. TBI is another example where a recent study showed that damage to the structural connectome can actually cause decreases in metastability of the brain's activity patterns. And that was also correlated with worse cognitive outcomes. And then in depression and disorders of consciousness, people have shown that there's a stronger relationship between structure-function coupling. So there's a stronger overlap of those two. And the worse it is, it's related to sort of more severe outcomes after disorders of consciousness and worse depression. So it seems that divergence from the structural connectome of the functional connectome is in some ways better than a collapse, say, of the functional connectome to the structural connectome. Some of the first work in my lab looking at structure-function coupling was really just a statistical analysis where we took each row of the structural connectome and correlated it with each row of the functional connectome. And so on a region-level basis, we got how aligned that region's structural connectivity was to the rest of the brain with that same region's functional connectivity to the rest of the brain. And this was analyzing HCP-YA, the young adult data. So this is about 1,000 individuals between the ages of, I think, 25 and 36. And we found that the structure-function coupling was the highest in strongly structurally connected visual and subcortical regions and weakest in limbic and default mode network regions. And it was consistent across time and in different sample populations. We also looked at using the structure-function coupling at a region level to predict things like age and sex and cognitive scores. And we found that this coupling was associated with age in certain regions. It was generally stronger in males, and that stronger structure-function coupling in the right insula and weaker coupling in bilateral middle cingulate were related to higher total cognition scores. And we also showed that it was highly heritable. So this sort of structure-function alignment, because of the familial structures in HCP-YA, you can look at the heritability of it. And we showed that it was highly heritable more so than just looking at functional connectivity or structural connectivity alone. So let me tell you a little bit about the network control theory work we've been doing. So this is sort of based around the idea that the brain is a complex networked system that can be controlled. And so you can use principles from network control theory and analyze, sort of apply them in the case of the brain. We know that our neural systems change their dynamics to meet task demands. That's how we can complete tasks and respond to our environment. And so that allows us to sort of have all of these plethora of cognitive processes. And more research is allowing us a window to see what neuroimaging substrates may sort of be the mechanisms of this cognitive control. And so we have this sort of idea that the brain can move between these kind of states of activation, and that the network control theory can be applied to see how much sort of energy or control energy is needed to move between different states and different activation states in the brain. So you can think of it as The brain activity, if it was reduced to a sort of 3-coordinate system, you could think of the sort of brain states as being these sort of clusters of activation that the brain sort of cycles through when you're just sitting there in the scanner during rest. So all of the analysis I'll show you is during rest. And you can basically just do a clustering of the activation of all of the time points over all of the subjects. You put everything into a big pile and you sort of do some clustering analysis and identify these commonly recurring states of brain activity. And so then you can sort of, from the time series of activation, you can get for every person, every scan, a time series of states that tells you which state the person was in at each of those TRs within the fMRI scan. And from that, you can quantify things like fractional occupancy. So how many frames, the percentage of frames that the person stayed in one state versus another. You can calculate things like dwell time. So how long did the person stay in that state, the person's brain stay in that state when it achieved that state, and things like appearance rate. So how many times per minute does the person cycle to this state? And then you can calculate things like transition probability. So how probable am I to transition from one state to another? So a lot of work has been looking at how this sort of dynamics, metrics of the sort of dynamics of the system change with different diseases and disorders. What our lab is focused on a lot is looking at network control theory, transition energy. So it's basically a way, this is the equations here are in the bottom right-hand corner. This is the differential equation that where X is the activity and T is time. So it's a vector of brain activity over time. A here is the structural connectome. So it's basically network diffusion plus some external input or internal input in the case of sort of task demands. And the control strategy is in B. I won't touch on that too much, but essentially it's a way of us determining how much control energy is needed by the system to complete state transitions. And so we've analyzed a lot of different data. One of the first applications that came out of our lab it was in psychedelics. So we analyzed acute people who had placebo or LSD, and they were under the influence of it at the time of scanning. Then we also had psilocybin data and DMT data, which was concurrent EEG and fMRI data. And in all of those cases, we see that psychedelics have a dramatic shift in the dynamics to where you need less energy to complete state transitions. And there's a concurrent increase in the entropy of brain activity signals. So that was kind of one of the first sort of applications in our lab. We also showed that sort of the region level control energy was related to the 5-HT2A receptor density, which is what the psychedelics are known to act on, the main actor for psychedelics. We also showed in HCPYA, in the HCPYA data that there's a subsection of the subjects who have heavy alcohol use, either alcohol use disorder or evidence of binge drinking. And we actually showed that global transition energy was higher in those subjects that had heavy alcohol use versus people who didn't drink. We also showed in MS that there's in early MS, when there's not a lot of disability, but there's some pathologies in the brain, the transition energy actually decreases. And then over time, as disability gets worse and the disease progresses, the transition energy actually increases. So we think that it might be reflective of possibly some kind of compensation mechanism followed by a degradation of that compensation mechanism. We also showed in sort of longitudinal study of people with mild TBI that it appears to be increased in subacute to chronic mild TBI, but that is kind of a smaller study. So we want to do a bigger study with a bigger population. And then we've also shown that this network control theory and transition energy is related to ADHD, but in a sex-specific manner. So it appears that the neural basis, at least using this network control theory, in ADHD might be different, varied between males and females. We also found a sex-specific effect of family history of substance use disorder on transition energy, wherein females with a family history of substance use disorder appear to have increased transition energy compared to females without a family history. And the opposite was true for males. So there seemed to be some sex-specific effects. And unsurprisingly, the transition energy at a region level is related to female sex hormones. So this was using the 28andMe data set. So we found that there were some positive relationships between transition energy in various regions and estradiol levels. We also replicated that in our aging cohort, where we saw that in the HCP aging data set that there was a positive relationship between estradiol and transition energy. So the higher estradiol, the higher the transition energy. And this, of course, we know aging mediates both of these things. So we found that that's true, but we did a mediation analysis and showed that the impact of age through global transition energy mostly goes through this estradiol effect. So it seems that estradiol and global transition energy are linked in some way. So what if we just completely forget all we know about the biophysics of the brain and just focus on using data-driven approaches to map between modalities? So that's where Keith comes in. So he's here. I'm glad he's here on this call today because this is his baby. And also he's like the best maker of GIFs, I think, that I've ever seen. Anyway, so maybe some of you saw that on Twitter, but in any case, the Krakencoder, it's the best name ever. So one of the sort of things that we were always sort of annoyed by when we're doing the structure function mapping is that there are various pipelines that can be used to extract functional connectivity and structural connectivity, and nobody knows which is the best way. So generally what we do in our lab is we replicate results using different atlases and fingers crossed, like hope that everything aligns. But in this work, what we thought we could do is perhaps sort of combine all of the different pipelines we could think of, all of the different atlases we could think of, and try to sort of put them all in the same framework. So of course, for functional connectivity, you have different choices like global signal regression, whether to use Pearson or partial correlation. Then there's things in structural connectivity world. So we can either do deterministic tractography, which is on the right here, which is a bit sparser and maybe perhaps less false positives. And then we have probabilistic tractography, which has less false negatives, but has more false positives. So there's sort of negatives and positives to everything. And we kind of are of the opinion that everything has some inherent information in it. So why not, try to use them all and combine them all together to get a comprehensive representation of the brain's networks. So I like to think of it like the sort of, I think it's an elephant in the dark, and everybody has a different part of the elephant and they're touching a different part. And then when they put that all together, they can tell that it's an elephant. But if you're just touching one part, you might not be able to tell. So I think that this is a kind of a nice way of combining all of this information that we extract through neuroimaging different pipelines. So again, here, yeah, so this is a sort of a schematic of all of the different flavors that we have. We have functional connectivity flavors, atlas flavors, structural connectivity flavors. So here's a representation of it where you have this autoencoder. So it's a shared autoencoder where you have encoding arms, or sort of, if people don't know about encoding and decoding autoencoders, essentially what it is it takes the data in and it sort of compresses it down to a lower dimensional representation. And then it has a decoding arm that allows you to reconstruct the original data. So the autoencoder itself is really about compressing data and doing a dimensionality reduction in the latent space of the autoencoder. And so here we have encoding and decoding arms for each of the flavors of functional connectivity and structural connectivity. And we have 3 different atlas types and global signal regression, no global signal regression, and then using Pearson and correlation and using partial correlation for the functional connectivity. So each of the flavors has an encoding and decoding arm, and they all go into this shared latent space. And I'll talk a little bit more about that in a second. But that's why it's called the Krakencoder. I think you can see it sort of aligns with the arms of the Kraken, sort of combining it into this common latent space. So here's a sort of schematic of all of the loss functions that go into training the Krakencoder. And if, like I said, for each of the sort of flavors, we have a encoder and a decoder. And then they have this shared latent space. You also can encode and decode across different flavors. So that's one of the tools of one of the applications of the Krakencoder, is sort of translating between flavors, mapping structure to function, but also mapping functional connectivity with global signal regression to functional connectivity without global signal regression. So all in, we train every pair of encoders and decoders in the strategy that we used. And then I think one of the sort of innovations that Keith came up with was making this shared latent space where he has a contrastive loss component, where each of the flavors of the functional and structural connectivity from a single person, they're all pushed together. So we want that all of the latent space representations from all of the different flavors of the same subject to be pushed together into the latent space and pushed away from other people. So one of the other things I haven't really talked about yet, but what was central to the Krakencoder was that we wanted to produce a tool that would do structure to function mapping and function to function mapping that preserved individual differences. So we really want to focus on not just reconstructing something that's highly correlated with the outcome or with the prediction, because then if you predict the mean, it's all highly correlated with the each individual's functional connectivity. But we wanted to really focus on preserving the inter-individual differences. And so I think using this contrastive loss term in the latent space really helped us to do that. So it really helped us to get this individual level differences when we do the reconstruction. So here's a sort of to sort of flesh out the details of the loss functions that we used. We have the path loss, we have the reconstruction accuracy, and the intersubject dispersion, and then latent space sort of contrastive loss, and then yeah, the latent space intersubject consistency. So I'm not going to go into too much detail, but I wanted to put it up there in case people look at the slides at a later time. There's also a preprint out on bioRxiv. So we trained this on the HCP young adult data. So this was 958 subjects. We all know the sort of demographics of them. And then we had also we applied sort of, I'll show in a minute, but we applied the frozen trained Krakencoder that we used, that we trained with the HCP-YA, and we applied it to the D and the A just to see if we could get out of sample sort of validation accuracy. So the performance metrics, I touched on this a little bit. We want to preserve that individual differences in the predictions. So we focused really on looking at identifiability, which in our case is basically looking at sort of subject by subject inner product of the predicted upper triangular part of the predicted matrix. And what we wanted to show is that subject by subject inner product has the highest, when you look at the rows, that the highest value is in the diagonal, right? So you want to be more correlated with your prediction. You want that prediction to be more correlated with your own observed connectivity versus everyone else that's in the test set, right? So you want your subjects' predictions to be as close as possible to their own data and as far as possible from everyone else so that you can get identifiability, perfect identifiability out of the predictions. And so that sort of top one accuracy, we call it, is for how many rows of this subject by subject matrix is the diagonal the highest. And then, of course, you might not be top one, but maybe you're top two. That's still pretty good. So then we want to look at the average rank of the diagonal within each row. So that gives us kind of a more robust measure of how well we're ranking in our identifiability metric. We also wanted to look at the sort of prediction in a sense that wasn't just identifiability, but how well are we actually predicting the values? So here we look at average correlation, or that's, I think, a little bit misleading because if you predict the mean, you get pretty high correlation with everybody's sort of functional connectivity if you're predicting functional connectivity. So what we did in the end was we removed the mean from the prediction and from the true value, and then we correlated the residual. Okay, so it's really like removing that effect of the mean and allowing us to not just predict the mean. And so our first result slide here, this is the top one accuracy for the Krakencoder for the test set that we had held out after we had training and validation. And then we had this really highly structured test set of 196 subjects. Out of those 196 subjects, we had 39 test-retest. So I don't know if everybody knows, but there were like around 40 subjects in the HCP-YA that came back another time 4 to 6 months after their first visit to get another scan. So that allowed us to look at test-retest reliability. And then within that 196 test set, we also had monozygotic twins, dizygotic twins, siblings, and unrelated individuals. So it allowed us to really look at how well we're doing with our prediction versus how well, let's say, using a monozygotic twin as my prediction would do, right? So that's sort of the baseline metrics are at the bottom, where we have sort of how well we would do if we used the population mean to perform identifiability. And of course, it's terrible. But if we use our dizygotic twins as our predictions, we get pretty good top one accuracy, and actually just about as good as the Krakencoder does for predicting functional connectivity from structural connectivity. I'm sorry, I should have really told you about this slide before I went into it too deeply. But this is the top one accuracy for every pair of encoding and decoding arms in the Krakencoder. So the encoding arm, the source, is listed here. These are all the different flavors of functional connectivity. These are all the different flavors of structural connectivity. And then this is just the column average and the row average on the final column there. And so you can see that predicting different flavors of functional connectivity from functional connectivity, another flavor of functional connectivity is actually pretty good. Identifiability, so we have top one accuracy that's at times 100%, but not really less than 67%, which is pretty good. Same for structural connectivity, a little lower there. So we still see that we have pretty good top one accuracy for predicting structural connectivity flavors from other structural connectivity flavors. But then, of course, as you expect, we know that structure and function, as I said, they don't really align perfectly. So predicting functional connectivity from structural connectivity, which is here in the bottom left-hand corner, and predicting structural connectivity from functional connectivity in the upper right-hand corner are a little bit lower, right? But we still have pretty high accuracy. You can see the highest flavors there are doing just about as well or better than dizygotic twins. So using my dizygotic twin as my prediction. So it becomes a little bit more a stronger case if you look at the average rank percentile, which again is sort of the average rank, the percentile of the of where the diagonal was when you kind of rank them by highest to lowest within their correlation with other subjects. And so you can see the average rank percentile for function to structures for certain flavors is about 99%. Same for structural connectivity, and that does just about as well as test retest. So using myself to predict myself at a different time, or my monozygotic twin to predict my other monozygotic twin. So it's doing pretty well in the sort of average rank percentile as well. Again, so using the sort of d-mean to average correlation, which is a lot lower than if you just correlate sort of the prediction with the prediction with the mean inside of it to the observation with the mean inside of it. And so you can see here it's a similar pattern where the function to function do pretty well, structure to structure do pretty well, and then you do have a drop, but it's still pretty good, still doing as well as a dizygotic twin would be for predicting them their own twin. Now, one of the sort of features of the Krakencoder that I really like are this multi-flavor fusion idea. So you have these different sort of pipelines to process in, let's say, 9 different flavors of functional connectivity and 6 different flavors of structural connectivity. So you can project all of these into the latent space, and they all have their own spot in the latent space, but you can then average those to get a single representation. And this is what we call the fusion flavor. So here's just a kind of a schematic of it where you have the different projections into the latent space and then the average there represented with the green dot. And from this fusion representation, you can actually do a prediction of all of the different flavors back out, right? So you can get projected in, do an average, and then project back out. And when we use this sort of fusion and sort of averaging in the latent space, we actually see that the top one accuracy gets even better for predicting between modalities. So here there's the fusion SC, so it's using like the structural connectivity from all the different flavors to predict the functional connectivity flavors. And we see we do a lot better with our top one accuracy than we did with the individual sort of atlases. Same for fusion functional connectivity and structural connectivity. And then the average rank is pretty high as well. So it gets even better when we have this sort of fusion representation. So our next question was, how well are we predicting specific networks? So are we predicting some regions or some networks really well and others not as well? So we wanted to look at the sort of the spatial distribution of accuracy and how that aligned with sort of the reliability of the measure itself. And so actually here we have Yeo networks representation of the accuracy. So they're sort of combined into these 7 cortical Yeo networks plus subcortex and cerebellum. And then we averaged sort of the accuracy of that either within or between network prediction. And so you can see that the fusion has this sort of pattern where you see a little bit brighter in the upper left-hand corner in the lower order regions, and perhaps a little lower in the limbic system and the subcortex. And this is actually pretty close to what we see if we take those 39 people who had a test-retest reliability and we just correlate their 2 measures. We see a kind of a similar pattern. And down below, we actually correlated these matrices with one another. And you can see that it actually aligns our Krakencoder predictions. The pattern of their accuracy across the brain aligns pretty well with the reliability of the metric of the measure itself. So it seems like regions or parts of the brain that have have lower SNR are also not predicted as well, which is sort of straightforward in the end. And then this is just sort of a reasonable representation of the Yale networks on the previous slide. We also wanted to see and make sure that the Krakencoder wasn't just doing a good job at identifiability and maybe the d-mean correlation, but that it was also preserving the topological properties of the networks. So here what we did was we computed sort of 4 different commonly used topological metrics for the graphs that were reconstructed using the Krakencoder. And we correlated these metrics from the predicted and observed connectomes across the test population. And we saw here that the Krakencoder graph metrics actually were correlated with the observed about as well as the monozygotic twins with each other, or the observed test versus retest with each other. So we're doing pretty well with preserving topological metrics as well. So finally, I wanted to compare against sort of these. We found really two ones that we could reproduce and sort of use the same training and validation and testing procedures so that we could have an apples to apples comparison with these 2 previous works. So this was a paper from Sarwar in 2021. They used a deep learning model to predict functional connectivity from structural connectivity. So that's the usual flavor, is predicting functional connectivity from structural connectivity. And here it was using this sort of deep learning network, and they had a penalty for sort of other different subjects correlations. So they tried to penalize like a high correlation between all of the predictions, so to try to sort of preserve individual differences a bit. And then there was this other paper in 2022. This is Neudorf, and they used a graph neural network to predict functional connectivity from structural connectivity. And here we trained and tested them and validated them using the same data. So this is a busy slide, but again, we have our top 1 accuracy, our average rank percentile, our d-mean correlation, and then this is the average correlation with the mean. And then the top 3 rows are the Krakencoder, and we tried to be as fair as we could. So we used deterministic structural connectivity or probabilistic structural connectivity or our fusion structural connectivity. And we compared them against the DeepNet, which is the Sarwar, and the GraphNet, which is the Neudorf, for the deterministic and probabilistic tractography. And you can see that across the board, the Krakencoder does a better job at sort of preserving the identifiability of the subject, as well as sort of getting that prediction accuracy. And then on the bottom, we have the topological sort of metrics and how well they were preserved. And you can see again that the Krakencoder does a pretty good job of preserving those topological properties. So then the next thing we wanted to do was sort of break open the Krakencoder, do an autopsy, if you will, on the latent space, to see if there's sort of some interesting information or some interesting representations that are occurring. And this is one of the very first figures that Keith created from the latent space. He just did a principal component analysis of the latent space and into 2 sort of dimensions and then colored them by sex. And you can see that it's pretty well dividing them. So here, I think the blue is male and the red is female. So you can see this is a pretty good separation. So there does seem to be some information, at least about sex. We also looked at a t-SNE plot of this is the test set. And like I said, within the test set, there are some dizygotic twins, monozygotic twins, and siblings. And so we connected the related pairs, and you could see that the monozygotic twins are almost on top of each other most of the time. So they're these sort of red lines or dots because they're really close to each other. And then they have some siblings in green, they're a little bit longer, and some dizygotic twins are a little bit longer too. But it does seem like at least the monozygotic pairs are lining up pretty well in that latent space. So the next thing we wanted to do was sort of look at the heritability in a more structured way. So here we looked at sort of the similarity of the latent space representations across different familial relatedness categories. So we have unrelated pairs here, which basically have 0 similarity. Then siblings have a bit higher, almost similar to dizygotic twins, and then monozygotic are really close. And then of course the retest are also very close to it. And when we compared statistically the differences between the relatedness pairs, we saw that there was significantly larger separation in the latent space compared to the sort of PCA projected functional and structural connectomes. And then we also used that latent space and we applied a kernel ridge regression model to do predictions of things like sex, age, and cognition. And sex and age seem to be pretty well predicted by the raw data itself, but when we get to cognition, we actually get a significant boost in the predictive accuracy using the latent space representations versus the raw data itself. And then I'll quickly go over these so Keith can do his tutorial, but we also then took the frozen Krakencoder and applied it to the HCP developing data and the aging data. So we didn't do any retraining at all. We just applied it out of the box, and we actually have pretty high average rank percentile and D-mean correlation for both the developing and aging populations, which is pretty surprising because they are very different aging populations. So the developing population is 5 to 21, and the aging population is 36 to 99. So it's very different from the data that was used to train the Krakencoder itself. We also did some sort of predictions of the accuracy. We do see that the familial structures are again preserved, but in the developing and aging populations, the predictions of the cognitive scores in age and sex seem to do better in the raw data there. So there might be some hint that in the future we could potentially retrain the Krakencoder to include information about developing and aging populations to make it more representative of those two populations. So if people are interested, there's a bioRxiv paper, and we're working on revisions now. So hopefully it'll be released soon, but it is released in preprint. So if people want to use that, they can. I also want to mention to this crowd, because I have many roles, as a lot of professors do, but one of them is as the co-director of the AI core for the Women's Brain Health Initiative. And so we're actually putting together some data challenges for women's brain health specifically. The first one will happen starting in August, September, and they're open to anybody. The first one is really directed towards university classrooms. And that one will be in the fall. And then we'll have an open public challenge for women's brain health, sort of a analysis of brain imaging data, predicting outcomes. And we're doing that with Kaggle and with Women in Data Science, WIDS. And so it'll actually have like prize money and everything. And so I invite you all to participate, and you can email me if you're interested in learning more. So really quickly, I just wanted to sort of get over this really quickly so Keith can take over. But essentially, this Krakencoder framework allows us to fuse multiple modalities and pipelines of processing into this common latent space and sort of do predictions of all of them, or use them all to sort of create this unified representation of the connectome, the functional and structural connectome. And it appears that we can use this in out-of-distribution data without really losing much accuracy. And using this in the future in sort of disease or disordered populations might allow us to look at how structure and function relationships may change with different diseases and disorders. So with that, I'll stop, and I can either take questions. I don't know how you want to do it, or maybe Keith can walk through the tutorial, or...
Maybe we can take questions first on your part and then go to the tutorial. Is that okay?
I'm just going to close my blind because my son is...
So let's see, what questions do people have? Oh, there's lots going on here.
Yeah.
Okay.
So I posted, by the way, in both the general chat and the Krakencoder chat, the Slack chat, there is one file missing for the demo to work, and Keith is going to demonstrate how to do this. So basically, you're going to need to copy and paste the code so I posted and run it in a notebook. I think Keith will demonstrate this.
Okay, what's happening in general? Okay,
Yes.
there it is.
Any questions?
I have a question.
Sure.
Maybe I'll kick this off. So one thing I was wondering while seeing sort of this strong separation of sex is that usually, when building classifiers of this kind, we can account pretty well for confounding factors, right? Like head size, things of that sort. And here in this framework, it seems like it would be really, really hard to account for those kind of confounders. But maybe I'm wrong. I might actually be misplacing my skepticism. So maybe, how do you think about confounding factors and how those fit into these kind of really data-driven things where with the data, all the confounders go in?
Yeah, so that's a good question. I've thought about this a lot, and I think I would say one of the things that we haven't checked is how it may be reflective of confounding information, but you could potentially take the latent space and say predict mean framewise displacement and see how much explained variance you could get in it. That's not necessarily like the best thing to do, and it's not like totally straightforward, because if you can predict it, doesn't mean that it's not useful. But yeah, but that could be definitely one thing we should look into. And you could even do it sort of in, we've talked about this before, Keith and I have talked about, so everything we presented right now is static functional connectivity, but of course there's dynamic functional connectivity as well. So you could potentially look to see if there was an effect of, like, let's say motion on within a certain window on a person's prediction and in the latent space of using their dynamic functional connectivity as well, to sort of get a track of how framewise displacement might impact it. I would say head size might have minimal effect on the network level analysis, but it would be good to analyze that for sure. And the other thing is too, like we've discussed, also having an arm for whatever we want to predict, right? So we could potentially retrain the Krakencoder with a demographics arm or a cognitive score arm that would help shape the latent space so that it might be better reflective of whatever behavior we're trying to predict.
Other questions so far? Okay. I don't see any questions either here or on Zoom. So maybe Keith, do you want to take over and share your screen and walk us through the tutorial? We'll probably need the font to be larger. Zoom in there.
You did. Okay. Can you see my screen right now?
We can see your screen, but the
Yes.
text is really, really small.
All right. I'm on like a big old external monitor. You know, I'm gonna unshare. I'm gonna just put it on my laptop and then maybe that'll be more normal. Okay, and share. Okay, can you see my screen now?
Yes, it's a lot better, but yeah, good.
I can
Maybe zoom in just once. Yeah.
okay. Okay, so can people see generally?
Yeah.
Okay. So you said this was the first sort of hands-on one. I guess I don't know how people have already worked through how to get on some of this, but if they haven't, well, let me copy this before I do it. But yeah, apparently some file disappeared this morning, but yeah, if you go into like the home folder here, there should be a curriculum folder, and then there's a Kusieski Krakencoder, and there's two in here for silly reasons, but the Krakencoder _ demo is the notebook that we actually want to run through today. This is, I guess, walking through what Amy sort of described. It's all on GitHub here. There's a link at the top of the notebook. So for more, try to move this header out of the way. For more detail on this, obviously you can follow this link. This talks, oh yeah, that's the file. We have a lot of examples and descriptions and, you know, ways to go through the data, another notebook that loads things. So other information is available. But we're going to just try to run through it and basically take a pre-trained Krakencoder model, the Krakencoder, which is the one that we have been training on the HCP data, and apply it to everything else. We're just going to take that pre-trained Krakencoder model, going to load it, and then we're going to apply it to some of the test data and then sort of see what those predictions look like. And as Noah was mentioning, there was a file that sort of got missing. So somewhere in here, but before this line runs, you need to copy and paste the line that was in the Slack there that's going to download that file. So I'm going to run that. That's just doing the little download, putting that one little file where it needs to go. Then we're going to run the second part, and all this is doing is it's running these little fetch scripts that in this case won't actually do anything because these files should already be on the image here. But if you're running this on your own machine, or if you're running this in like Google Colab, this will go and fetch these data files that are needed to run the Krakencoder and download them and put them in the place they need to, and then load them up here. So now we have the files that we need. This is the checkpoint. It's like a PyTorch model checkpoint that has all the weights. And as Amy mentioned, all the inputs go through a PCA transform that's been pre-computed on the training data. So we have to download those as well so that we can apply that to new data. So now here, this line, I'll just go through cell by cell. It's pretty straightforward. This is just going to load in the checkpoint. Oh boy. Oh, I think that's just a warning. That one's fine. These didn't happen yesterday, so I don't know what changed in the meantime. Here we have the transformers, which in this case, these are these PCA transforms, and we're just going to load them in. We've fetched them, but we're going to load them in, reading in these PCA transforms. Here is where we actually create the model object. No, nothing exciting happening now. Now we are going to load in some data. Now the way that data can be loaded in a few different forms. The form, and this just has a little widget here. If I run that, it brings up a little widget, and it has a little file browser. And the way that we're reading in most of the data here is as a sort of, I'm calling it a bids-ish zip. So I don't know if you guys have had discussions about any bids related stuff yet. It's something I'm sure you guys will encounter. It's this, sort of standard for all kinds of neuroimaging data and formats. And it's great for connectivity data. The standard is not finalized yet. And so the files, the formats that I have here is sort of an approximation of a bids tool here. This zip file, I'll just jump over to the terminal here. And just show what's in it. So the zip file contains a bunch of little TSV files, one per subject per flavor. So it's got this subject, this atlas, and this is the flavor, partial correlation high pass filter. So we have a lot of files in here, obviously, and a little participants info file. So we click on that zip file, we add it, and it loads in all those data files. So we have 20 86 by 86 SC probabilistic matrices, 20 probabilistic deterministic SC matrices. We're loading in all the data. This would be if we just hit run and it'll load in the same data, but without the fun GUI aspect. So the data as read in are square matrices. They're region by region connectivity strengths between regions. The way that the data actually gets processed in the Krakencoder in the sort of schematic here, the first step was, just bringing it up, was compressing the upper triangular, they're symmetric matrices. So we just take the upper triangular and squish it into a vector so that each subject is a vector, is a row in the overall matrix. So that's all we're doing here is just vectorizing, and it just tells you, okay, we went from 20 86 by 86 to 20 for this matrix size, it goes to 3,655. In this case, I'm only reading in 5 flavors for the sort of smaller atlas that has 86 regions. Amy mentioned we have 3 different atlases, but just for demo purposes, this was a lot quicker. So we squish this matrix size down into this matrix size. And then this step here, when you have, for this, it's actually a little bit unnecessary for this data because this is HCP, which is, the test data here is from HCP, which is the same as what we trained the model on. But for other data sets, like when we wanted to apply it to the HCP development or aging, because of differences, particularly in acquisition, also some in the demographics, you have different distributions, like domain shifts in your matrices. And if you want to apply a model to data that has a different distribution, you have to do something to be able to have it apply the same distribution that it's expecting. Here, we're doing a very simple approach for that, which is just take my input data. We can actually specify certain subjects in the input data that are considered training, but in any case, we'll take the mean connectivity for those training subjects and get just a vector. And then we just do a simple linear transformation to map from that single mean to the training mean that the model was trained on. So it's just a very, sorry, my kid just got home. Hopefully I won't hear it, but so yeah, we're just for each flavor, we just compute the mean of the input data and then just do a very simple, 2-parameter map to the training data. So I'm going to run that, and it's just telling you for each flavor. In this case, the 2 parameters were 1.004 plus, times x plus 0.003. So all of these are close to like 1 times x plus 0, because again, they're from the same distribution as the training data. So now we have the data that is ready to run into the model. And now we're going to take each of those flavors and we're going to put it into the Krakencoder model. In this case, I guess I did it a little bit. Everything's kind of a shortcut here. Hopefully it's commented enough to be able to tell what's going on. But basically, I'm looping through each flavor. And here, I'm just taking that input data that's been adapted just by that linear function. It's been adapted, and I'm running it through my network. And in this case, what I'm doing is I'm telling it which flavor is on the input arm. And then I'm telling it, in this case, decoder index -1 means don't convert it, like don't decode this output. So all I'm doing is taking it from input to the latent space and keeping it in latent space. I can here say go from input to latent to any particular output flavor. But here I'm just saying go into latent space. So now I have, for every subject, for every input flavor, I have the latent space representation for that subject, for that flavor. Sorry, I heard some sound. Is it just an echo? Okay. Okay. So what we're going to do here is, like Amy was talking about, we can create a fusion representation. Now you can take all the latent vectors from all the flavors and average them into sort of a unified fusion representation and then decode all the flavors. Just as an example here, I'm going to actually limit it. And I'm going to say, let's only average the data coming from the structural connectivity. So we've taken structural connectivity, we've mapped it into a latent space. Now we're going to just average those latent vectors to get a single latent vector per subject that it represents their structural connectivity in the latent space. And then I'm going to average them. So this encoded fusion in this case is just averaging the files that were, or the data from latent space. Now I'm going to take that averaged latent vector and I'm going to run it through the decoders to create all of the predicted flavors. So again, I'm looping through all the flavors. And in this case, I'm already in latent space. So my encoder is -1, meaning don't do any encoding, but now decode to each of the flavors that we have in the model. And here is the output that it's printing out here. It's just telling us our latent representation after we averaged it all together. There was 20 subjects, and it's 128-dimensional vector per subject in the latent space. And now we have taken that latent vector and we've put it through all the decoders. So now we have a 3655 vector for each subject for this flavor and this flavor and this flavor. And we also have, the model is trained to have all those other atlases as well, even though our input data only had the 86 by 86 matrix. So we have predicted connectomes for all these other flavors, higher dimensional flavors as well. So we have predicted connectomes now, and we have our input observed connectomes. We want to take a look at, this one's a sort of real just quick look at what those connectomes look like. I'm just going to run it. Hopefully it goes quickly. Yeah. So all this is doing is taking for just picking one flavor. This is the Pearson correlation for the 86 region atlas after high pass filtering. And I'm just going to look at one subject for that flavor. So the first subject in this case. And then what I'm going to do, first, I'm going to take my, if you remember, the inputs to the matrix were vectorized and the outputs to the matrix are also vectorized. So I just need to convert those back into a square matrix. So there's just a little function in here that's in the utilities for the package. But basically, it's just going to go through and take that row of the adapted data and turn it into a square matrix, and take that row from the predicted data, put it into a square matrix, and then it's just going to plot them. So we're just using matplotlib im shows here. And there's nothing There's nothing amazing about these figures. They're just, the values that are in the matrix. In this case, they do look very similar. That's good. We have some of the features we look for, like having this, in this case, the cortical ROIs are arranged first by left hemisphere, then by right hemisphere, and otherwise they're in corresponding order. So we have this kind of off diagonal band. So that just tells us, okay, good. We have the kind of data we're looking for in both cases. As Amy mentioned, in the true data, like the observed data, everyone is actually very similar to the population average. And this looks very similar to the population average. So even if I just faked it and just showed the population average over here, visually, these would still look the same. But I figured it was worth just having eyeballs on. Here's one of my inputs, and here's one of my predicted values for that. So now going beyond that, we have the question of how well did this actually do at predicting, my connectome versus someone else's connectome. So I'm going to actually shorten this. It's not really it just takes a little bit longer if it has 10,000 permutations. But so basically what I'm going to do here is, maybe we'll just move down to the first one. So this is for a predicted flavor. This is a probabilistic structural connectivity here. And of course, I was predicting it from the average of the encoded structural connectivity. Now I have a couple subject by subject matrices. I had 20 subjects in my input. So I have a 20 by 20 matrix here. This is the correlation of every subject's observed matrix and every other subject's observed matrix after removing the population mean. So if I didn't remove the mean, all of these would be like everyone's correlated with everyone else at like 0.9. But we've removed the mean. We have, of course, the identity here along the diagonal because every subject has a correlation 1 with its own observed. And then you have this distribution of the other values. This is the same thing, taking all the predicted connectomes against predicted connectomes. Again, the diagonal is 1. The off-diagonal values in this case are higher than they were for the observed. We do, anytime you're doing these predictive models, especially with the regularization that has to go along with it, I mean, there is a tendency for the predictions to be sort of, I mean, it's denoising. So it's sort of closer to each other than in the noisy input data, if you want to say that. Now, this is the sort of more useful part here. This is every row here is an observed connectome, and every column is the predicted connectome. And what we've done here is for every predicted connectome, we are just showing sort of as a line plot what was the value reflected in the colors here. So nothing groundbreaking there, but we are highlighting, in this case with the circle, which predicted subject had the highest correlation with that subject's true observed connectome. And in this case, we see exactly what we wanted, which was along the diagonal, you have the highest values. The diagonals are not 1 because these are not identical connectomes, but the value along the diagonal is always higher than the values for any other subject. And so in this case, the values down this side here are just showing me the rank of my prediction. So it's 1 out of 20, which is the best. So my average rank is 1. We reflect it usually in our paper, especially as a percentile, where 0 would be, or where one is perfect. And in this case, 0.5 is actually the sort of random chance. Here, we're showing a permutation test just demonstrating what that average rank percentile would be if you just scrambled all the rows. So in this case, 1,000 times, we just scramble the predicted rows, do the same analysis of basically where was my own row in the row of each observed connectome. And when you scramble it, you end up with this random distribution that, around 0.5. The value we had was, of course, 1.0, which is well above that distribution. It's not super surprising here because we did use the structural connectome to predict these structural connectomes. This is the deterministic case. Nothing really changed here. Here is where we are taking, again, structural connectome data. We're predicting a functional connectome. So this is a much harder problem. There shouldn't be a one-to-one correspondence, but the model has tried to do its best to learn the relationship. Here, again, the sort of predicted versus predicted and observed versus observed don't tell you anything that useful. If we look at the predicted versus observed here, you see a slightly different pattern than what we saw before. It's not perfect. It's not that every single diagonal was always the highest value in that matrix. So in this first one, for row 1, column 1 was the highest. So that's awesome. We have a rank 1 for that subject. For the second observed subject, the highest among all of these was row 2. So again, awesome. That has a rank of 1. Rank of 1. This one here, if you look, this one, it has a high value here, but it's apparently not as high as the correlation with this predicted subject. So instead of being a 1 here, this is the third. Sorry, I heard is that just an echo or did? Okay.
I think it's just.
If there are questions, I'd maybe ping me on Slack or something. I don't know. Hopefully I'll see them, but I'm happy to answer questions at the end as well. So in this case, this subject's prediction was the third highest out of the 20. In this subject, it was actually the second lowest, so that's bad. But if you look through all the rows and you take the average of those ranks, it's 4.2. And that's out of 20. Out of 20 subjects, the average subject was ranked 4.2, which is a percentile here from 0 to 1 of 0.84. So we have a 0.84 percentile. And again, the permutation testing here is clustered around 0.5. And so we're greater, p-value at permutations, at 1,000 permutations is less than, 0, essentially. Even with 10,000 permutations, is still 0. And then the same is true for the other connectomes that we're predicting. We have a average rank percentile of 0.818, an average rank percentile of 0.895. So again, it just helps to see that they're not necessarily exactly the connectomes that went in, but we are doing way better than chance. And as Amy showed, we're doing better than other sort of competitive models. And we're, if you did a similar computation where you took the monozygotic twin versus myself, you'd actually have a similar sort of top one or average rank percentile as well. So the last part here is just going to take those and take those matrices and put them back into the square form. So now we have our predicted where we had this, and now we create our square matrices again. That's so that if you want to actually work with them as matrices, I had done it up above, but this is just if you're running it and you don't want to run all that visualization script. This just produces a new structure here that has, 20 86 by 86 matrices for every flavor. And then finally, we're taking that data and we're going to save it as another, what I'm calling a bidzish zip file. And again, this just has a little GUI thing. It doesn't really matter, but if I save it, it's taking every subject's file, it's sticking it into a zip file and adding it to that zip. And again, you can kind of see the format we have here. We've got, subject and identifying information. This is just telling us it's a Kraken fusion prediction. And if you just open these, it's a TSV file, and you'd see it's an 86 by 86 matrix that you could use in any other, connectome processing pipeline or toolkit or whatever. So that is the whole rundown here. Hopefully that kind of made sense how we go through it. Like I said, there's a lot more information on the GitHub here, and I'm happy to, field any feedback or questions, or, if people have problems running it either on the Slack for the program here, or, GitHub issues or email, anything like that. So that is it. Are there, I don't know, were people actually able to get that going?
You've got a question on Slack, and I'll just read it to you here. So it says, so what I'm understanding is that you are separately creating a connectivity matrix in MATLAB and then pushing it into this coder. If this is true, is there a specific way the data would need to be prepared or created in MATLAB to work best with the coder?
So we don't actually use MATLAB in any of this data here. The connectome files that are input are just stored as TSV files that, different pipelines spit out, like the XCP that sort of takes fMRI clean outputs and parcellates them and does some denoising and stuff like that. or MRtrix will produce files similar to this. So really, as long as it's a region by region TSV file, they can be read in. In terms of the actual pipelines so if you're going to feed it, the way it's run right now, if you're going to feed in data to the Krakencoder, it does need to match one of the flavors that we trained it on. So it has these sort of 15 flavors. I already have that up, but it has these 15 flavors for these sort of 3 specific atlases, and we did sort of some specific denoising strategies. We used high pass filter instead of band pass filter, and some things like that. There's, like I said, some more details about those specific pipeline choices on the GitHub. But if you process it using those pipeline choices, one of those pipeline choices, then you can create a TSV file, which you can do in MATLAB, or in PyTorch, or in Python, any other tool, Excel, and then you can load it in and feed it in like that. Hopefully that makes sense.
As a hack project, somebody could pretend they're giving you some kind of structural connectivity, actually created some other way, I don't know, via some simulation or something like that. Give it to the Krakencoder and see what happens, right? That could be a fun exercise.
Yeah, exactly. And we have tried, we have used this on certain kinds of simulated data. Just we have a tool called the NEMO tool, network modification tool, that takes lesions and creates estimated structural connectivities. So it's sort of an estimated structural connectivity based on injury, and then fed it through this as though it was the real structural connectivity to see, what kind of functional connectomes it predicts and things like that. So it can definitely be used on, you have to tell it it's a pretend version of some flavor of connectivity that we've trained it on. But yeah, it's wide open as far as, feel free to throw random stuff at it and see what comes out.
And I will say that Keith is now working on adding arms to the Krakencoder in an easy way also. So different pipelines, you can kind of update it without retraining the entire thing. And it seems to work pretty well. So yeah.
So like one example, I mentioned we did all our functional connectivity was using high pass filtered time series. And that's one of the many choices that people make, is whether they're going to high pass filter or band pass filter, where they do temporal smoothing before computing the correlation matrices. We found the high pass filtering preserves a lot of useful information, but the whole point of this project was sometimes you have a different flavor that wasn't one of the ones that other people have been working with. And so you want to maybe simulate what that data would look like in another pipeline. So yeah, we've taken bandpass filtered data and trained an arm to go into the same pre-trained latent space as all the other ones. And so then the same can be true for like new atlases. So our goal is to eventually just have a big library of lots of different flavors that all go to the same latent space that's been sort of pre-baked by, we kind of picked for us anyway, a pretty exhaustive choice of atlases and processing choices to try to make it as general a latent space as possible. And, now we're going to be kind of baking in new arms to that latent space.
Yeah, JB.
I think you answered a little bit of that already, but if I wanted to actually, first of all, it was a lot of fun. It was great to see that happening. I mean, but if I wanted to specialize the latent space to some application, how easy would it be to actually tune that training for that specific application, but starting from the latent space that you have, and say, okay, I just want to tune it to that specific use case. Is that how easy would that be?
I didn't completely hear.
I'll repeat the questions. So the question was, let's say that we wanted to train this for a very particular application. Let's say we're trying to predict, I don't know, like reading ability in kids, right? How hard would it be to retrain the Krakencoder into something that does that? Like,
so
could you take this network and specialize it?
I guess I'll say two things. So one is you can take the without retraining the Krakencoder, you can train models that predict one of these features from the latent space as, you know, as it already exists. That's what Amy was showing with these sort of demographic predictions, cognitive predictions. That was just taking like the 128 dimensional vectors that had no, you know, explicit information about what the cognitive or age or whatever were, but just trained a linear predictive model to go, you know, from 128 dimensions to that predicted value. That's, you know, as easy as any multilinear, you know, regression problem. If your goal is to, like she was saying, maybe there's some room to train a new version of the model with explicit information to condition that latent space even better. And there, we are still thinking about the best way to do that, to making sure that you're sort of balancing, you know, if you have just a single dimensional feature that you're trying to predict, and you're training that as an arm against all of these, you know, 3,000 dimensional connectivity vectors, there could be some trade-offs that you have to play with in the loss functions or something. So we don't know the exact answer to that. But in theory, retraining is very doable with the package the way that it is. I mean, there's some example scripts in here to do the training if you have a set of connectomes. But for a specific demographic feature, like I said, you'd have to do more thinking about the right way to do that. And that part isn't exactly built into the package currently, but it is something that we're interested in fleshing out. Any other
question, JV? Yeah, follow
I just have a question on the loss function. I mean, obviously there's that part that actually makes the same subjects connect to, I mean, they're close to each other. That's one aspect of the loss, but it's not the only one. How do you weight, you know, that loss function for the various things you need to have on the latent space?
Did you catch that? Do you want me to repeat it?
Sorry, it's just a little, it's kind of echoey and distant there. So if you can pass that along.
So I'll just repeat it. I'll try to see if I can repeat it. The question I think is the loss function is really complicated, right? It has all these different elements to it. And then the question is, how do you know how to weight the relative contributions of the different elements of the loss function? Is this just empirical or?
Yeah, I mean, it's a obviously good question, and that's all these, like any model you have is going to have immediately sort of all these questions about how exactly you choose your hyperparameters, and you know, there's so many directions you can go. In this case, it was empirical. We sort of trained lots of different versions of this model with, you know, weights on each of these features ranging orders of magnitude, and you know, and even then it's still a trade-off. There's not even a single optimum there. Especially as Amy was mentioning, we have sort of the identifiability metric and the sort of correlation with the observed metric. And those are sort of inversely related. And so parameter choices that maximize the identifiability actually decrease the sort of correlation accuracy term. And, you know, it's kind of bias variance trade-off kind of thing. And so we kind of plotted a lot of versions of it in a multi-dimensional hyperparameter space, and kind of found a shoulder where it was kind of starting to us seem like the right trade-off, and then that's the one we went with. But that is a huge open question in all these models, is how exactly you decide to select your parameter weights.
If you want to look at the figure, it's in the supplemental information on the preprint on our bioRxiv.
Oh, that's right. Yeah. The bioRxiv link is also on the GitHub. I can pull it up. So yeah, and in the supplement, there's that kind of figure, I think, that I'm talking about, where it's like the identifiability versus the correlation. You kind of have this line dropping off as one dimension of the weights is shifted, but there's all other dimensions as well. Any other questions?
Other questions from Zoom, from the room? If not, then we'll thank Keith and Amy again for. their
Thank you so
presentation.
much.
Thanks for visiting with us.
Well, thanks so
That
much for
was so
having us.
fun. Thank you.